Hello, everyone. Welcome to our webinar. Today, we're presenting identification of long-range allosteric changes in the Nipah viral protein G using the Biode delta, presented by Biode and Joyce Wong, PhD. I am your moderator, Gabriel Mercado. Welcome again, everybody. Ligand-induced conformational changes in biomolecules are usually solved by arduous experimental or very specialized technical equipment. Also, the knowledge these analytical tools provide is usually limited to a structural snapshot from which functional insights cannot be easily derived. Recent incorporation of the second harmonic generation technology, or SHG for short, into the biophysical toolbox has allowed scientists for the first time to routinely identify dynamic behaviors and test their molecular mechanisms models in an economical, accessible, and straightforward way for the first time. And that is what we are going to talk about today. Joining me to do that is Stanford scientist Joyce Wong, from who we are going to hear present in a moment. But before we get started, I want to take a quick moment to talk to you about how you can interact with us today. It's very important for us to hear your questions and maybe engage in a discussion about not only Dr. Wong's results, but also about questions you have about the technology. Our GoToWebinar viewer has two parts: the viewer window, where you will see today's content, and the control panel, where you can interact with us during the presentation. You can submit your questions at any time by typing in your questions or comments using the question pane on the control panel. We will answer as many questions as we can during the live Q&A, but questions about the technology itself will be answered afterwards via email. The ones we don't have time to address will also get a written response after the event. The control panel will automatically hide itself to give you more space for the viewer window, and you can open and close the control panel by clicking on the arrow button on the panel tab. Now, a little bit about our speaker. I am very excited to present to you Joyce Wong, who has been a structural biologist at Stanford since 2012. Together with Professor Ted Yardesky, she has focused her research around the characterization of the paramyxovirus, fusion and attachment proteins mechanism, a critical step that initiates the highly pathogenic effect that these viruses have on humans, and for which no vaccines or therapeutics are currently available. Joyce, thank you very much for being here, and over to you.
Thank you. So today I'll talk to you about the allosteric changes induced in the Nipah virus attachment protein G, induced by binding to its cellular receptor, affinity 2. So some background about the Nipah virus. It is a negative sense single-stranded RNA virus in the paramyxovirus family, and is a prototype member of the Nipah genus. It was first discovered in 1998 when transmitted from farm pigs to humans in Malaysia. However, the natural reservoir for Nipah virus is Pteropus fruit bats. Nipah virus causes severe disease in humans and is considered a BSL-4 organism. The mortality rate in outbreaks is 40 to 92%. It causes respiratory symptoms, and its most dangerous clinical manifestation is encephalitis. There is currently no specific treatment or vaccine approved for human use, and periodic outbreaks still occur, mostly in Southeast Asia. Therefore, we wanted to study how this virus enters host cells and causes infection. Nipah virus, like other paramyxoviruses, is encapsulated in a lipid envelope that contains proteins that mediate entry into cells. These proteins are the fusion and attachment proteins, indicated here in a schematic of the Nipah virus genome. So in our current models of paramyxovirus entry, the fusion protein F inserts a fusion peptide into the host cell membrane and undergoes a large-scale conformational change. The attachment protein is a tetramer consisting of a globular domain and an alpha helical stop. The head domains bind a receptor on the host cell. Currently, evidence points towards the fusion protein F and the attachment protein G of Nipah viruses associating prior to receptor contact. Upon binding to a receptor, which in the case of Nipah virus is F1, B2, or B3, the globular heads of G are thought to move from a heads down to a heads up position, exposing stock residues that contact F and trigger its fusion function. So in work published by Liu et al. In 2013, Nipah-G was shown to undergo stepwise conformational changes in response to a dimeric construct of F1, B2, F1, B2, Fc, which is thought to represent the natural oligomer of F1, B2. This protein is a chimera between the natural ectodomain of F1, B2 and a human IgG Fc domain. So these changes in conformation in Nipah-G are detected by conformation-specific antibodies. So there is a decrease in binding to the conformation-sensitive antibody MAB215, and an increase in binding to another antibody, MAB45, and this finally culminates in F triggering. So in our study, we wanted to know what is the conformation of the Nipah-G tetramer, and how does this conformation change upon binding to F1, B2? And thirdly, is dimerization of F1, B2 necessary for Nipah-G triggering? In order to visualize the conformation of Nipah-G, we perform negative stain electron microscopy. What are shown here are the 2D class averages of Nipah-G and their distribution from 2 rounds of classification. A representative class average is shown on the right. These show that Nipah-G does form a tetramer of globular head domains around a central stalk. From this, it can be seen that the structure of Nipah-G is different from structures of the attachment proteins from other paramyxovirus that have been previously solved. There is a substantial separation between the apical dimer of globular domains and those bound to the stalk. Also, there is a gap between those at the mid portion of the stalk, meaning that they are monomeric, while those of Newcastle disease virus and parainfluenza virus 5 all form dimers. We were unable to image Nipah-G in complex with F1, B2, Fc due to aggregation. In that case, we turned to different techniques to study conformational change in response to F1, B2. And one of those techniques was second harmonic generation, or SHG. In our experimental setup, purified Nipah-G ectodomain protein is bound via His6 tags at the membrane proximal end of the stalk to an INTA head group containing supported lipid bilayers. The sample is illuminated by a laser that creates SHG at the bottom of the well. Signal from an SHG active dye covalently conjugated to Nipah-G is then monitored over time. The SHG signal is sensitive to both changes in the net orientation of the dye relative to the surface normal and to the shape and width of the conformational distribution that may occur due to changes in protein dynamics. Therefore, two aspects of ligand-induced change are detected by this technique. We used SHG to characterize Nipah-G mutants thought to represent different conformational responses to F1, B2 binding. These mutants are localized to 2 regions on the Nipah-G globular head domain. Region 4N contains L181A, while region 9 contains K376A, C387A, and Q388A. These mutants were previously characterized by binding to the conformational antibodies mentioned earlier, MAB213 and 45. As a refresher, wild-type Nipah-G undergoes a decrease in binding to MAB213 and increase in binding to MAB45 upon addition of dimeric FMB2. In addition, binding to an antibody raised against the Nipah-G stop, polyclonal antibody 167, increases. The mutants fully characterized by these antibodies are C387A, which undergoes overall less binding change, and L181A, which appears to be pre-activated in a conformation that exposes the MAB45 epitope, even without FNB2FC addition, and it undergoes little further change when FNB2 is added. So in our experiment, the Nipah-G ectodomain constructs were labeled with SHG active dye at nonspecific lysine residues by standard NHS ester chemistry. The proteins were labeled to a similar degree of labeling, about 1.7 dyes per Nipah-G monomer. Some lysine residues were more predominantly labeled than others. The main ones are K84 and 130 on the stop, and K201, 246, and 386 on the globular head domain. For the most part, the distribution of labels among the lysine residues is similar between mutants. So here are the time courses of SHG signal change over time following injection of FNB2FC into wells of immobilized SHG-labeled Nipah-G. Readings were taken every 5 minutes over a range of FNB2FC concentrations. It can be seen that there are differences in the degree of Nipah-G signal change between the mutants. This is most notable with K376A and Q388A, where the net signal change appears to be greater upon equilibrium. In C387A, the signal change is minimal until addition of extremely high FNB2FC concentrations. Mutants can be more directly compared by calculation of delta SHG at a set FNB2 concentration and time interval. In addition to the differences mentioned earlier, L181A shows a significant increase in the amount of SHG signal change compared to wild type. Next, we showed that these changes are specific to the Nipah-G FNB2 interaction in a series of negative controls. FNB2 was incubated with unlabeled soluble Nipah-G prior to addition to SHG-labeled Nipah-G containing wells. This resulted in minimal signal change. This means that there was competition by unlabeled Nipah-G for FNB2 binding. Similarly, incubation of the endogenous receptor for FNB2, EPHB2, prior to addition led to a decrease in FNB2 binding to Nipah-G. Also, minimal SHG signal change was observed with wild-type Nipah-G and any of the mutants upon addition of Ephrin A1-FC, which is a non-binding isotype of Ephrin. We next measured the binding affinity of FNB2FC to each Nipah-G mutant by ELISA, in case differences there explain the differences in SHG signal. Nipah-G was immobilized to NINTA-coded ELISA plates, followed by addition of FNB2. The KD for most Nipah-G mutants was similar to that of wild type, which was 88 picomolar. The exception was C387A, which had greatly reduced binding affinity and aberrant, inconsistent binding curves that prevented measurement of KD by direct binding assay. Therefore, a binding inhibition assay was designed instead, where FNB2FC was mixed with either wild-type Nipah-G or C387A prior to addition to wells containing bound wild-type Nipah-G. the C387A mutant was defective in competition for efferent B2FC binding to the immobilized wild-type Nipah-G, meaning that the small signal changes in SHG in the C387A mutant were most likely due to lack of binding. Next, we tested all of the Nipah-G ectodomain constructs with wild-type binding affinity for how their binding to conformation-specific antibodies changes upon efferent B2 addition. We wanted to see if the behavior of the ectodomain was consistent with that previously observed for the full-length protein. Nipah-G was bound to NiNTA-coated lysoplates, followed by incubation with efferent B2 and MAB213 or 45. Overall, the changes in binding to MAB213 and 45 were similar to those previously reported. The wild-type protein again undergoes a decrease in MAB213 binding and increase in MAB45 binding. Notably, L181A does show an elevated level of binding to MAB45 relative to wild type, even prior to efferent B2FC addition. Also, there is a less drastic change in MAB45 binding with increasing efferent B2 concentration. So mutants that were different from wild type in the shape of their MAB binding curve also have differences in HHG signal. There is one exception, Q388A. So MAB213 binding could not be measured with this mutant because this mutant destroys a key epitope. The MAB45 binding curve of Q388A is practically the same as wild type, but the SHG signal change is different from wild type. This could mean that SHG is detecting differences that cannot be detected by monoclonal antibody binding. These changes may be in the dynamics of the protein and not just in net conformation. And this will be described more in the next part of the talk. Due to the differences, like the difficulties in imaging the Nipah-G efferent B2FC complex, we imaged Nipah-G in complex with a monomeric form of efferent B2 in the hopes that they would form single particles, and they did. 2D class averaging of Nipah-G complexes with the monomer did not reveal obvious changes in overall conformation or conformation distribution. A representative class average is shown where additional density for the efferent B2 monomer can be seen on the lower two globular domains of Nipah-G. There is a range of conformation for the pairs of globular domains with respect to each other, and the two extremes illustrated here are designated the aligned and cross conformations. So both are present in Nipah-G with or without efferent B2. So we compared the effects of dimeric efferent constructs to two monomeric constructs. So these monomeric constructs are the full ectodomain of efferent B2 up to residue 229, as well as only the globular domain of efferent B2 up to residue 167. So the lack of apparent conformational change in EM was further supported by binding of conformation-specific antibodies to Nipah-G in the presence of increasing amounts of efferent B2. So dimeric efferent B2 addition causes a difference in the binding to MAB213 and 45, similar to what was reported previously. However, binding to these two antibodies did not significantly change with increasing amounts of the monomeric constructs of efferent B2, even at concentration ranges adjusted for their higher KD, which was measured to be about 10 nanomolar. Given the lack of apparent conformational change with the monomers, SHG revealed a surprise. Monomeric constructs of FNB2 also induce SHG signal change. There were differences in the delta SHG between all three FNB2 constructs. The robust response of the monomeric construct suggests that changes in the protein dynamics rather than large-scale net conformational changes are occurring. We performed hydrogen deuterium exchange mass spectrometry to investigate the monomer-induced changes further. The basic concept is to look for changes in the amount of amide backbone hydrogen exchanged with deuterium. Changes in peptide regions may occur due to occlusion to the solvent upon ligand binding and/or changes in the dynamics of the protein. So the regions with most significant difference in hydrogen deuterium exchange are mapped onto the crystal structure of the Nipah-G head domain in complex with monomeric FNB2. So this illustrates that the changes are occurring in regions even distant from the binding interface of FNB2 to Nipah-G, meaning that allosteria is taking place. The regions with the most decrease in exchange are colored blue, and they are regions which directly contact the FNB2 monomer. The regions with most significant increase in exchange are two regions, one in the globular domain and one in the stalk, and these are colored in red. Residues 253 to 266 and 321 to 337 form a contiguous region on the opposite side of the FNB2 binding interface. Regions 82 to 110 and 128 to 140 are part of the predicted alpha helical stalk domain. Now, I will summarize the data from these multiple techniques in a model of Nipah-G activation by FNB2. Our data adds to the existing model by describing allosteric changes induced by monomer binding that occur prior to full activation by dimeric FNB2. Upon binding of one efferent globular domain, Nipah-G changes in its dynamics, and this is detected by hydrogen deuterium exchange mass spectrometry and SHG. But this does not cause large-scale conformational change, as observed by EM. These changes make up an allosteric signaling pathway from the efferent binding interface of the globular domain to the stalk. The dimeric nature of FNB2 allows the second head domain to bind with increased avidity. This binding results in further conformational changes that are detected by differences in binding to MAB 213 and 45, and this leads to exposure of the stalk so that G can activate F fusion. So I would like to thank the members of the Jardetsky lab and my supervisor, Theodore Jardetsky. For the work with SHG, I would like to thank Tracy Young and Joshua Salasky from Biode. For the electron microscopy work, I would like to thank Jiayan Zhang and Shihang Liu from Hangzhou's lab at UCLA. And I would also like to thank Elizabeth Comidis at UCSD for the work with hydrogen deuterium exchange mass spectrometry. And thank you.
Thank you, Joyce. We have already some questions on queue, but please let me remind the audience that you can enter your questions at any time, and we'll get to as many of them as we can. Okay, let me start with a question by Gina Rinetti. Gina Rinetti is actually asking me, after your conformation-sensitive antibodies failed to show you an indication of a big conformational change, why did you go to SHG and not directly to deuterium exchange?
Because we were already working with Biode, and it was the most accessible at the time.
Would you say that SHG is more accessible than deuterium exchange?
At the time, yes.
Very well. I have another question here by Ben Moore. He wants to know if the current assay that you have for this virus would allow you to produce a drug screen to look for inhibitors that stop the infection mechanism of this virus.
Yes, I believe that this current SHG assay would allow you to do that. So we basically have an assay now to look at conformational change induced by receptor binding, and I can conceive that you could screen drug molecules that would block the SHG changes that result from FNB2 binding.
I see. Okay, let me see. I have another question here. This is from Chicago. It says, could you have obtained a sense of the long-range conformational changes that are occurring on your receptor with other methods like fluorescence?
Yes, I believe so. But the challenge with fluorescence-based methods, at least as I understand it, is that it is necessary to modify the protein with fluorophores, and trying to find a place where it can be modified without affecting the native protein activity is a challenge and needs to be carefully screened for.
Okay, this is another question I had. Urethrin B2 has an FC part that you didn't test on your assay, but the FC part seems to be required for the full elongation of your receptor to produce the infection. What do you think the FC is contributing? Does it also bind to the to your receptor, for example? Is that known?
So the FC part in our FNB2 construct is not part of the native protein. It is a chimera between the native FNB2 ectodomain and the FC domain of an IgG antibody. So the purpose of it in this assay is to form an artificial dimerization domain. On the native, in the native context of ephrin, it is a membrane-embedded molecule where it does have a transmembrane domain that would mediate weak dimerization at the membrane.
Okay. I have here another question by Svetlana at UCSF. She wants to know, can fast kinetics be measured by a biodesi? What is the dead time of the instrument before the time course of the signal is measured? I think I can answer that question better because it's an instrument question. Yes, the fastest the biodesi can go right now is about 2 seconds. So as soon as you inject anything on a well, you can have read on that well every 2 seconds. And this is enough to be able to distinguish the slow conformational changes from the very fast conformational changes. Okay. Any more questions from the audience? If no, I have another question here. Let's see. How important is the fusion mechanism for the paramyxovirus infection? Has this infection step been validated in other viruses? I guess what they are asking is, has it been shown that if you interfere with this first step of the virus recognition, do you stop infection? Do you know anything about it?
Oh, are you referring to the attachment protein interaction with this receptor, or the fusion, which is mediated by the F protein? Because both are required.
Yes, I guess both. The first one is before the elongation of the fusion protein is the recognition of the receptor to the virus, right? Has anyone ever interfered with that and stop infection?
For the Nipah virus, I believe there is a monoclonal antibody out there that binds directly to the Ephrin B2 binding site, and this has been shown to inhibit infection.
Okay. I have another technical question here. They're asking, how long did it took you to develop and apply an SHG assay for the first part of your project? Was it fast, or did it took several months?
I would say it was relatively fast compared to other methods that I have tried. The main limitation is actually the time it takes to produce the protein and to get access to the instrument. I think as long as you have those, developing the assay and optimizing the conditions can happen fairly quickly.
Very well. Okay. We seem to have run out of questions. Please, if anyone in attendance has any questions, this would be the time. Okay, I have one more from Omar. Omar is asking, is modulation of dynamics as opposed to modulation of binding or conformational change a viable therapeutic option? Omar is at Berkeley.
So in our assay, it's just an in vitro assay to look at the effects of receptor binding on the protein. As far as its therapeutic value, that I cannot predict at this point. But this is a starting point to look at possible effects that compounds might have on the protein, and that can be validated in a more clinical setting.
Thank you so much. Any more questions from the audience? Okay, if not, thank you so much. We have exhausted all the questions our audience has had. So I want to thank everybody in attendance for his engagement. My thanks to Dr. Wong and Biode for making today's webinar possible. And if you join us late or would like to share this with a colleague, we have recorded this webinar and will soon send out the link to the recording. Thank you all for being here. On behalf of Biode and Dr. Wong, this is Gabriel Mercado saying thanks for joining us.